traditional herbal medicine research. So before our guest speakers talk, please let me introduce the agenda of this sub-panel in bilingual. 尊敬的各位观众，尊敬的各位嘉宾，大家晚上好，欢迎大家来到分论坛3传统草药研究中现代科研方法的应用。在嘉宾开始演讲之前呢，请允许我用中英双语为大家介绍分论坛的流程。Our guest speakers, Dr. Luo, Professor Zheng, and Dr. Zhou will share with us their research and their understandings of traditional herbal medicine. 我们的3位嘉宾来自中国中医科学院中药研究所的骆路博士，来自上海中医药大学的郑月娟教授，和来自上海中医药大学附属龙华医院的周忠年博士，会依次向我们介绍他们对传统中草药的见解和研究。After their lectures, we will start the discussion section. 在3位嘉宾演讲之后呢，我们会进入问答与讨论环节。Please feel free to submit your questions in chat. We can use either English or Chinese. 请您在 Zoom 的聊天窗口把您想要问的问题打在公屏上，中文或英文提问都可以。If your question is only to one of the guest speakers, please specify their names. 如果您是对3位嘉宾中的疑问进行提问，请您在提问中明确指出。Our guest speakers will answer your questions based on their language preference. 3位嘉宾会根据他们自己的选择，用英文或者中文来回答问题。If you are not fluent in Chinese and you do not understand their answers, please feel free to send an email to hopkins.asian.medicine@gmail.com. Yifeng, would you like to type down this email address into the chat?
Yes.
在问答环节中呢，如果你有任何的问题，请您发邮件至以下邮箱，一峰会把邮件地址打在对话框里。Now let's welcome Dr. Luo to introduce the strategies for analyzing the efficacy and chemical composition of traditional Chinese medicine prescriptions by computational methods. 现在让我们欢迎骆路博士为我们分享通过计算手段分析中药方剂功效与化学成分的策略。
Can you guys see my screen? Okay. Hi everyone. Very glad here to share one of my studies about how did we do in silico analysis for herbal medicine that has been used for treating COVID-19. So I'm just going to briefly introduce some of the facts and data we have from during the pandemic, what's happening. So over 90% of the new COVID-19 patients, wrong. Just a moment. So I'm gonna introduce my research based on the following 5 aspects. First of all, the comparison between traditional medicine and Chinese medicine regarding the methodology of herbal medicines and the drug use in the clinic. So during the pandemic, what's happening in China back in 2020 and the late 2019, over 90% of the patients in China, as well as in the original outbreak place in Hubei province, has conducted herbal medicine. Our China Academy of Chinese Medical Sciences has also sent a clinical team to Wuhan to help in the clinic for treating patients during the pandemic. So in the basic theory of Chinese medicine, COVID-19 or the coronavirus was considered as deadly toxin, and in the etiology aspects. So it is combined with Japanese heat toxin deficiency and blood stasis. So generally speaking, it covers 3 categories in etiology: the Japanese, which means it's very chronic, and the toxin, which means it changes a lot, it has a lot of variety, and it is a pandemic, so it's infectious. So the targets in basic theory of Chinese medicine, it targets at the tie-in foot meridian of spleen, as well as the tie-in hand meridian of lung. And the principle for treatment would be to dissolve the dampness and remove the toxin. So in a basic theory of Chinese medicine, targets at both the lung and the spleens. Those are the 2 organs that would like to be attacked by dampness, according to Big's theory of TCM. So in clinic, COVID-19 was classified into generally 4 stages: the mild stage, the severe stage, the critical stage, and then recovery stage, or death. So during the mild stage, both dampness and the toxin are accumulated in the lungs, or dampness and toxin are accumulated in the spleens. And when it develops into the severe stage, it will close the lungs and spleen, because in basic theory, lungs and spleen are in charge of transformation and transportation of the nutrients of our body. If it loses the function of transformation and transportation, the nutrients or the external etiology factors will attack our body and accumulate into dampness and the toxins in the body. And when it develops further into critical stage, there would be the mechanism would be Qi deficiency involved with blood stasis, which would damage the yin of the body. And during the recovery stage, both lung and spleen Qi will be deficient, or it develops even more critical into death. It will be the depletion of yang and yin, and which involves multi-organ damage. So in Western medicine, the clinicians have been here, have told us it shows both symptoms from SARS and MERS, which are both coronavirus affecting lungs. It involves bronchitis and COPD symptoms as well. When it develops further, spleen and the immune system will also be damaged, which involves spleen dysfunction and lymph cell decreasing. When it goes further, it will involve heart, kidney, and the liver, as well as multi-organ dysfunction. So in February 2020, a group of scientists in China published an article on Lancet about the symptoms and the data that connected regarding the immune system, which signifies that the lymph cells, white blood cells, as well as CCR4+, CCR4/6, Th6 and 17, all those signals that are showing the immune reaction are increasing. So when we talked about like the multi-organ dysfunction, so heart was involved, especially when the toxin in Chinese medicine or the virus attacks on the cardiovascular system. A lot of immune reactions will be triggered, which will also include the fluid immune reactions. Most of death cases indicate that the heart function was damaged as well. So knowing to all that SARS-CoV-19, the second virus was a beta single RNA virus. So the spikes, which is S, will bind to the ACE protein to attack to the host cells. So ACE could be expressed in multi-organs, including lungs, bronchi, nasal cavity, intestines, kidneys, and testicles. So once the immune reactions were triggered, the cytokine storm would be triggered, which is the critical mechanism that helps the syndrome to develop into critical stage. So during the cytokine storm, a lot of immune factors would be increased, as for example, CD4 plus, CD8 plus, IL-6, all those immune reactions are getting worse. The president of our Academy, China Academy of Chinese Medical Sciences, their team have used a variety of Chinese patents represented by Lianhua Qingwen capsule, and which involves a lot of herb studies removing toxin and heat, which we will discuss about later. This is a case study published by the team of academician Zhang Boli. So they have found that like the disappearance time of the symptoms, the recovery of the body temperature, and hospitality days have been decreased if the regular treatment are combined with herbal medicine treatment. So this is the article we have published as a cover story in APSB. Generally speaking, look at this figure. There is a Tai Chi yin yang at the center. So which kind of perfectly corresponds to like the immune system, because immune system we also have upgrading and downregulating immune actors, and also in and yang we have evil chi and healthy chi fighting against each other, which can correspond to antigen and antibody in immune system. So we know that in biomedicine or conventional medicine, the categories of infectious disease were classified according to the pathological microbes, including mostly there are 5 categories: virus, bacteria, fungus, chlamydia, and mycoplasma, and parasites. Well, in traditional medicine back during the classical biblical Chinese medicine, infectious diseases, just as the same as other diseases in Chinese medicine, were classified according to the symptoms that are showing on patients. So they have classified the 5 pathogens into cold pathogens, worm, larynx infections, and parasite infections as well. So traditional medicine is more like symptom oriented, while biomedicine or conventional medicine is more like pathogen oriented. Here is a more specific figure explaining that. So in biomedicine, for treating infectious disease, we are trying to first of all classify the different pathogens, including virus, bacteria, and fungus. Then we try to diminish the pathogen, like antivirus treatment, antibacterial treatment, antifungus treatment with a lot of modern technology, and we're trying to regulate the immune system together with a lot of immune regulators. While in traditional Chinese medicine, because it's more like symptom oriented and it's more focusing on the holism, the whole body to regulate the symptoms. So it is more targeting at the symptoms, while when we prescribe a description, we use the 4 tiers of monochlor, minister, messenger, and advisor in Chinese, it's 君臣佐使, to treat the symptoms. And especially for infectious disease, the theory in basic Chinese theory is that they would attack our body from 6 layers, including Taiyang, Yangming, Shaoyang, etc. The 6 layers gradually from exterior to interior. And the syndromes and the stages will develop as the pathogen attacks the interior layer of the body. While for herbal medicine, when we are doing research, we are trying to dissect or analyze the pure compounds or molecules to find out the mechanism that is hitting, that is causing the symptom relief function. So we're trying to find data from the clinic, which is also a kind of evidence-based medicine or research. So especially for my research, we have connected 25 protocols that was officially published by Chinese governments in different provinces for treating COVID-19. We have connected 166 modified herbal formula as well as 179 single herbal medicines. Then by using in silico analysis, we found from all of them, we got 8 core combinations, and we developed 10 new formulas, as well as we had analyzed on their therapeutic effects, chemical components, and medicinal properties, among which those medicines are the most frequently subscribed during the COVID-19 treatment. They both have functions as antipruritic, expectorant, and cough suppressing, and as well as damage resolving. So first of all, we filtered a lot of chemical components. Eventually, we found 1,212 chemical components that are included in our database for further cluster analysis. So at the table and the right table are the most frequently used, the top 10 most frequently used herbs during COVID-19 in China. So like 甘草、黄芩、苦杏仁, those are very representative, like antipruritic herbs, as well as heat and toxin removing herbs. So again, we analyzed the therapeutic facts as well as the medicinal locations, whether it is roots or fruit or leaves or the whole plant. Then we selected the most frequently appeared chemical components among the over 1,000 molecules. And then we did network analysis of the core herbal combinations and chemical components. Eventually, we found there are 8 core combinations, which are the basis of our future new prescription developments. So the focus of our study is self-organizing mapping. Eventually, it in distinct dispersed structure similarity. We eventually got 12 core clusters with ring circles. Here it is showing the 12 clusters with ring circles. And those molecules, the 12 clusters we found, 8 of them have ring circles. And then we did in silico on the platform, and we found a lot of metabolomic targets that are involved in the immune system. And eventually, we did unsurprised hierarchical clustering to find new 10 new prescriptions based on the 8 core clusters with the ring systems. So based on this research, we proposed a new methodology for in silico analysis on natural products like herbal medicine but not limited to herbal medicine. So the classical methodology is to extract chemical components within a piles of herbal medicines and trying to do in vivo, in vitro, and clinic trials to prove its therapeutic effects and mechanism, which is time consuming and money consuming. However, if we do from the bottom to the top, it's like if we do, so thus figure B is showing the classic from the top to the bottom classic method we do in herbal medicine. However, if like what we did for the herbal medicine for COVID-19, if we first filter the molecules on computer by in silico analysis and SOM, then we can find core combinations as well as clusters that are having mutual effects and targeting at the immune system. Then based on those findings, we can withdraw the numbers of herbal medicine we have to extract chemical analysis. So it will save us a lot of time, effort, with a lot of modern biotechnology like deep learning and machine learning, as well as SOM. So this is the methodology we proposed in our research. So when we were drafting the figure for the most frequently used herbs and clusters in of herbal medicine in COVID-19, we got the idea like the yellow golden groot is a symbol of herbal medicine tradition in Chinese culture, even though it is very traditional with a lot of plants, leaves and fruits and everything. We can isolate all those molecules with modern technology like cluster analysis, SOM, as well as network analysis. So the virus maybe can be expelled. So our article was chosen as the cover story in 2019 by APSB. So that is all my sharing. Thank you.
Thank you so much for your sharing, Dr. Luo. 非常感谢骆路博士的分享。We saw some questions in the chat. We will help you record your question and then ask those questions again in the Q&A session.
Screen. Can you see my screen?
Yes.
Chinese medicine has been used for cure disease for thousands of years in Asia. It also contributed a lot to human health. Until now, and lots of modern techniques or most are applied for the research and development of Chinese medicine. Today, I would like to show you a part of my research work related to Chinese herb medicine using zebrafish model and RNA-seq technology based transcriptome analysis. My talk's title is Zebrafish Swimming to Our Site for Anti-Intracellular Hemorrhage Drug Discovery from Chinese Herb Medicine. My presentation includes the following 5 parts: research background, experimental design, key results, conclusions, and publications. Firstly, I will make a brief introduction about zebrafish. Zebrafish is a tropical fish. You can see its picture on the left bottom of the slide. As its name characterized, zebra chopped tool could be found on the whole zebrafish body surface. It is very small, and the length of adult zebrafish only 3 to 4 centimeters. But the genetic similarity is more than 80% with human. This genetic similarity makes the experimental result in zebrafish much more convincing in human. In addition, zebrafish has a high fecundity. One pair zebrafish could produce 300 to 400 eggs every week. Zebrafish also has many other advantages. For example, lower cost, short experimental period, and particularly the transparent amber and the tissue-specific fluorescence transgenic lines makes the observation much more easier under microscope. Thus, our group construct various disease models in zebrafish. For example, cerebral hemorrhage model using vessel-specific green fluorescence and blood-specific red fluorescence, double transgenic zebrafish line. Toxin-induced Parkinson's disease model indicated by locomotion dysfunction and neural loss after TH staining in the zebrafish brain, pro and anti-angiogenesis studies using a transgenic zebrafish line FLA1, and the heart-specific green fluorescence transgenic zebrafish was used for the research of chemotherapy-induced cardiotoxicity, and the T-cell neutrophil or macrophage specific fluorescence transgenic zebrafish has been applied for research focused on the regulation of Chinese herb medicine on inflammation and immunity. All these above models have been well established and used for the related drug discovery from Chinese herb medicine and underlying mechanism study. Today, I will demonstrate an example and progress of zebrafish model used for drug discovery for the anti-intracerebral hemorrhage from Chinese herb medicine in our laboratory. So what is intracerebral hemorrhage, which is abbreviated to ICH? ICH is also named hemorrhage stroke. It's the second most common subtype of stroke after ischemia stroke. It only accounts for 10 to 30% of overall stroke. However, compared to ischemia stroke, ICH leads to high mortality and more severe disability. The case fatality rate of ICH is 40% at 1 month and 54% at 1 year. Less than half of the patients could survive after suffer from ICH, and only 12 to 39% of survivors can achieve long-term functional independence. ICH accounts for half of the disability-adjusted life years lost due to stroke worldwide, and dramatically destroyed the quality of patients' life and caused great burden to our society. The unmodifiable risk factor of ICH includes old age, male gender, Asian ethnicity, while the modifiable risk factor for ICH includes arterial hypertension, current cigarette smoking, excessive alcohol consumption, hypocholesterolemia, and prescription medications, and so on. Among them, hypertension is the most related risk factor, and the management of lowering blood pressure dramatically decreases the incidence of ICH, particularly in developing countries. The number of medication-caused ICH increased in the recent years, such as anticoagulations, statins, and COX-2 inhibitors, and so on. The case of oral anticoagulation-associated ICH increased because the oral anticoagulation drug is an important medication of atrial fibrillation in older, and the prevalence of atrial fibrillation significantly increased along with the elevation of older population. Moreover, some clinical studies revealed that high dose of statins also increased the risk of ICH. Unfortunately, no individual treatment has been shown definitely to improve the outcome of ICH. So, how does ICH damage our brain? Let me introduce you the time course and brain injury during ICH. When the cerebral vascular ruptures, the blood bleeding into leukemia and forms hematoma. The hematoma expands in the first few hours, and the hematoma expansion further results in mass effect in the brain. The mass effect of the hematoma leads to the deformation of normal brain structure and compress both brain tissue and cerebral vessels, which significantly impairs neural function and cerebral vascular barrier function. So, hematoma expansion is a leading cause of early deterioration. Then the edema occurs around the hematoma, which is called the perihematoma edema, and further contribute to the mass effect and increase intracranial pressure. Finally, the hematoma is resolved and cleaned through the red blood cell lysis and erythrophagocytosis. In addition, the hematoma also could be evacuated by surgical interventions for elevation of mass effect, edema, and resolution of hematoma. But randomized control trials did not show benefit outcomes of surgical intervention compared with medical management in patients with ICH. So ICH causes direct injury from the mechanical effect of hematoma and edema, while the secondary injury includes following several aspects. Firstly, the osmotic effect of the clot causes movement of steroid into surrounding brain tissue. Secondly, the production of thrombin dramatically causes inflammation in the brain. Thirdly, the iron release during the process of red blood cells lysis and leads to neurotoxicity. Last but not least, the blood-brain barrier is disrupted by the cytotoxic edema. In summary, ICH dramatically result in brain injury due to the mass effect, inflammatory effect, and the cytotoxicity of the hematoma. Current treatment method of ICH mainly target to the syndromes and complications in different progression stage of ICH. For example, lower the blood pressure at the initial of ICH because the elevation of blood pressure is one of the basic physiological reactions when hematoma occurs in the brain and cause further hemorrhage. The pharmacological treatment for the reducing hematoma growth could be used in the stage of hematoma expansion, and the intervention promotes the cleanup of hematoma, such as surgery or disruption of intraventricular hematoma by alteplase. However, no specific therapy directed towards one aspect of ICH progression has been shown to improve outcomes of outcome and survival rate of ICH on the basis of randomized control trials. But all these treatment decreases the rate of death in hospitalization. Patients who underwent maximum medical and surgical management have bad outcomes. So the understanding of how ICH happens and prevention from ICH is very important. As we all know, ICH is caused by the rupture of a cerebral vessel and not a single entity. 85% of cases are due to cerebral small vessel disease, predominantly deeper perforator atrophy and cerebral amyloid angiopathy. Deeper perforator atrophy is also termed hypertensive atrophy or atherosclerosis, and cerebral amyloid angiopathy is caused by Alzheimer's disease. And the disruption of vascular integrity is the same pathophysiological characteristic. Vasospecific architecture contains 3 layers. The outermost layer mainly consists of connective tissues. The middle layer mainly consists of vascular small muscle cells, which controls vascular contraction and relaxation. The inner layer mainly consists of a single layer of endothelium supported by a subendothelial layer of epithelial tissue. And endothelium mediates the interchange of mass between tissue and blood. It is vital for the maintenance of vascular function and integrity. Various ICH-related diseases such as hypertension, atherosclerosis impair the vascular integrity via the disruption of vascular endothelial integrity and function. So vascular endothelial integrity and function plays a critical role in the control of vascular integrity. Moreover, vascular endothelial cells tightly connected together and form a single layer of endothelium. And vascular endothelial integrity is dramatically regulated by cellular contractility, permeability, and cell-cell junction, particularly the touch junction and adhesion junction in endothelial cells. So in summary, ICH dramatically contribute to the disruption of vascular endothelial integrity, and protection of the vascular endothelial integrity might be promising in the treatment of ICH. Traditional Chinese medicine has been used for treatment of disease for thousands of years, and silver materialism, which named Danshen in Chinese, is one of Chinese herb medicine presented a potent vascular endothelial protective effect. Danshen is a widely used Chinese herb medicine with the function of promoting blood circulation and removing blood stasis in traditional Chinese medicine. Danshen is mainly treatment for the cardiovascular and cerebral vascular disease in clinics, and exists multiple pharmacological activities, including antioxidant, anti-inflammation, and anti-apoptosis. Several commercial available drugs are developed from Danshen and approved by China Food and Drug Administration, such as compound Danshen pills, tensional capsule, and sodium tensional 2A sulfonate injection. So Danshen might not only present significant vascular protective effect, but also a good safety profile. According to previous phytochemistry studies, Danshen mainly contains lipophilic constitute, including tensiols and terpenoid, and hydrophilic constitutes such as Danshen Su and silvanolic acid. In my study, I proposed the cerebral hemorrhage protective effect of Danshen, as well as the discovery of its promising active ingredient and underlying mechanism study. Traditionally, the ICH animal models is constructed by injection of bacterial collagenase or autologous blood into brain in rat or mice. In my study, we employed an emerging ICH animal model in zebrafish. As previously described, zebrafish is belongs to vertebrate and has many advantages, and it is suitable for high throughput drug screening. HMGCR is a rate-limiting enzyme in the production of mevalonic acid, and mevalonic acid is the main substrate for synthesis of cholesterol. Advastatin, which is also named Lipto and belongs to statins, is a commercial available drug to lower cholesterol by inhibiting the activity of HMGCR. Several clinical trials revealed that the high dose of atorvastatin elevated the risk of ISH. In our lab, we established an atorvastatin-induced zebrafish ISH model to mimic the rupture of cerebral vessels in a transgenic zebrafish line, which express green fluorescence protein in vascular endothelial cell and red fluorescence protein in red blood cell, and used for the discovery of a drug candidate for treating ISH from traditional Chinese medicine, Danshen. Here is my experimental design. Firstly, the dancing extract was prepared by water extraction, and the commercially available constitute of dancing were also collected. The double transgenic zebrafish line was employed and generated vessel and blood-specific fluorescence amberes. Atorvastatin was used for induction of cerebral hemorrhage in zebrafish and co-incubated with dancing extract and its constitutes in multiple well plate. Then the cerebral hemorrhage protective effect of dancing extract and its constitute were observed under an epifluorescence microscope. So the compound presented cerebral hemorrhage protective effect were picked up and followed by further drug efficacy evaluation and underlying mechanism study in zebrafish in vivo and vascular endothelial cells in vitro. Here I will introduce you the main method used in my study. Zebrafish was employed as the in vivo model to evaluate the efficacy and underlying mechanisms of cerebral protective effect, protective compounds. Firstly, we generate a double transgenic line of zebrafish by pairing of a single transgenic zebrafish line. This new generated zebrafish line increased the sensitivity of observation of red hemorrhage in the zebrafish brain with a black background on the epifluorescence microscope. Secondly, the OD staining was used for the indication of hemorrhage in zebrafish brain. Hemoglobin is a main content of red blood cell in hematoma and presents peroxidase activity. It can transfer the black format of reduced OD to the brown format of oxidized OD. So the OD staining of zebrafish further confirmed the efficacy of active constitute of tension on atorvastatin-induced cerebral hemorrhage. Thirdly, the recovery of neural function is important in ICA treatment. In my studies, neural function of zebrafish was measured by locomotion test. After drug treatment, the zebrafish were distributed in a 96 well plate, and their swimming distance was real-time tracked in the zebra box. The total swimming distance of zebrafish reflect its neural function. Fourthly, the RNA-seq-based transcriptome analysis contribute to the elucidation of the underlying molecular mechanisms. After drug treatment, the mRNA of each group was extracted and followed by cDNA library construction and sequencing. After sequencing, the data was management by bioinformatic analysis. Finally, the proposed targets or signaling pathways were verified by the real-time PCR and classical pharmacological experiment. Here comes the key results. Firstly, we established the atorvastatin-induced cerebral hemorrhage model in zebrafish. Zebrafish were treated with various concentration of atorvastatin for 24 hours. Then photographed by a fluorescence microscope, we found that atorvastatin significantly increased the hemorrhage rate and hemorrhage area in a concentration-dependent manner, and the hemorrhage rate is above 80% over the concentration range of 2 micromolar. Then the Danson extract and its lipophilic and hydrophilic constitutes were co-treated with atorvastatin for 24 hours. We found that dancing extract significantly decreased the hemorrhage area. Also, the hemorrhage rate is not decreased. And the similar result were also found from Danshensu and SAA. Moreover, SAA decreased both hemorrhage rate and hemorrhage error, and tension 1 dramatically decreased the hemorrhage rate in a concentration-dependent manner, regardless of other indicated tensionals. So we could conclude that SAA, SAB, and Danshensu might be the main water-soluble constitute, which presents a cerebral hemorrhage protective effect in Danshen, and tension 1 might be the main lipophilic constitute, which presents a cerebral hemorrhage protective effect in Danshen, and the tension 1 is promising for the ICH treatment. So we verified the cerebral hemorrhage protective effect of co-treatment tension 1 in double transgenic zebrafish line. And first evaluate its cerebral hemorrhage protective effect by pre-treatment. Zebrafish embryos were pre-treated with tension for 2 hours, tension 1 for 2 hours, and washed out and treated with atorvastatin for another 24 hours. The hemorrhage rate and error were further evaluated by OD staining. We found that tension 1 significantly decreased both the hemorrhage rate and hemorrhage error over the concentration range of 303 micromolar. Another compound presented promoting cerebral hemorrhage protective effect was sodium tensional 2A sulfonate, which is abbreviated to STS. STS is a water-soluble derivative of tensional 2A. Tensional 2A is the main lipophilic component of tension. However, tensional 2A not decreased the hemorrhage rate in zebrafish as previously described, but we found the STS dramatically decreased the hemorrhage rate in a concentration-dependent manner, and the treatment efficacy is above 90% over the concentration range of 10 micromolar. In addition, the toxicity of STS is much lower than tensional 2A. STS not present any toxicity up to 300 micromolar in zebrafish. Furthermore, we found that STS concentration dependently decreased the hemorrhage error in double transgenic zebrafish line, and we also tested whether STS promote the recovery of neural function in zebrafish cerebral hemorrhage model. Zebrafish embryos were co-treated with atorvastatin and STS for 6 days, followed by locomotion testing. The data showed that atorvastatin significantly decreased the swimming distance, and co-treatment of STS increased the swimming distance. So we could conclude that STS significantly attenuated the extent of cerebral hemorrhage and locomotion dysfunction induced by atorvastatin in zebrafish. In order to explore the two underlying mechanisms of STS protecting against atorvastatin-induced cerebral hemorrhage, we also conduct a transcriptome analysis by RNA-seq technology in zebrafish. Zebrafish embryos were divided into 3 groups: control group, atorvastatin treated group, and atorvastatin and STS co-treated group. There are 40 zebrafish embryos in each group, and the independent replication is 3. After drug treatment, the samples were collected and followed by RNA-seq and data analysis. And we identified 207 and 346 different expression genes in control group versus atorvastatin treated group analysis, and the atorvastatin treated group versus atorvastatin and the STS treated group analysis respectively. And we also find 64 overlapping genes in these 2 groups of different expression genes. Furthermore, the expression level of 53 different expression genes, among which 5 genes were upregulated, and 48 genes were downregulated in the advanced treated group versus control group analysis were reserved in the advanced and STS co-treated group versus advanced treated group analysis. So this 53 different expression genes play important role in the underlying mechanisms of STS protecting from advanced increased induced cerebral hemorrhage. These 53 different expression genes were also subject to GOKGT and PPI analysis. And finally, we found 12 hub genes, 20 hub genes were involved in the protective effect of STS against cerebral hemorrhage and its vascular integrity promoting effect. They code proteins like hemoglobin, carbonic anhydrase, sodium hydrogen exchanger, and HIF1 alpha. The mRNA expression level of these proteins were further verified by real-time PCR. We demonstrate that the mRNA expression levels of hemoglobin, carbonic anhydrase, and sodium hydrogen exchangers genes were increased by outstanding treatment, and co-treatment of STS with outstanding significantly decreased the expression level of these genes. Thus, STS dramatically affect oxygen metabolism and exchange in advanced-induced cerebral hemorrhage. So we could conclude that Danson and its constitute presented a promising ICH protective effect. The order of ICH protective effectiveness was STS, tension 1, SAAB, Danson extract, Danson Su, and SAA. Hemoglobin, carbonic anhydrase, and sodium hydrogen exchanger genes might be potential biomarkers of advanced-induced ICH in zebrafish, as well as the pharmacological target of STS. Zebrafish is an attractive model for drug discovery from Chinese herb medicine and underlying mechanism study. New research techniques like RNA-based transcriptome analysis, along with classical pharmacological approaches, might contribute a lot to the elucidation of the action mechanism of Chinese herb medicine. The main content of my presentation has been published in 4 CI journals and 1 Chinese journals. That's all. Thank you very much.
Thank you so much. Thank you, Dr. Zhou, for your sharing. That would be all the presentation for today. And now we are going to move on to the discussion period. I know we are running late, so we are going to dive straight in. Our general procedure will consist with the same as what Cici mentioned. You may type in your questions to any of the speakers in the chat box, and we are going to refer those questions to the speakers for you. So now, our first question, I think, would be going to address both speakers who are still here. I think I really want to dive into this question about, the practice of molecular and cellular biological techniques, research techniques in the discovery of efficacy, clinical value, or even the molecular mechanism behind certain molecules, certain contents contained in certain herbal medicines. I want to ask you, so from what I heard was that from Professor Zheng's presentation, you mentioned using UHPLC, which is these 高效叶相色谱 to basically elucidate all of the contents within the extract, and then using classical bioinformatics or classical single cell research techniques, or for example, fluorescent microscopy, for example, PCR, for example, Western blotting, to identify the cellular mechanism or the cellular effect of a certain compound that comes in. I'm really intrigued by the idea of applying both, of finding really crucial like data points or basically getting how do I put this? Basically elucidating all the different molecules within a very complex herb. How can we establish that? Or are there more techniques that we can do that applies to both speakers? Because in Professor Zheng speaking, you mentioned a few drugs that you tested, and in Dr. Zhou's speaking, you mentioned danshin as the major drug. How did you guys come to the realization that the certain molecules are the crucial ones? How do we elucidate them from the rest? And how can we move forward to apply classical research approaches and treat them as like molecules that you will get from like a Western medical approach, basically? 我简单的用中文翻译一下，帮助同学们理解一下。我的提问主要就是我们如何从一个复杂的草药的成分当中，将所有的有效成分清晰的分离出来，并把分离出来的这些用相关性进行排序，意识到哪些是在这个它的临床疗效当中起到关键作用的。这个我们是如何实现这一步，并且如何把这一步很好的嫁接进现有的这个主要的现在的这个细胞，包括这个分子生物学的主要研究手法。
很好的问题啊，我可以用中文回答吗？是这样，你这个问题非常重要。因为首先我们说那个中药的作用，可能我们先去明确它里面有什么样的成分，这是我们利用化学手段是可以做到的。但是化学手段可以做到呢，它有2种方式，一种是定性的方式，一种是半定量的方式。它可以看到哪些化合物的分度是比较高的，所以这个综合几方面的因素来判断哪些化合物是比较重要的。比如说中医基础理论指导下的君臣佐使的这个使用，那么君药和臣药相对来说可能重要性会更好一点，更重要一点。另外呢，就是我们刚才也说了，它半定量的方式的话，就可以知道在入血成分里面，或者在肺脏，或者在心脏，你感兴趣的器官里面富集到的主要的化合物比如说前20位是哪些，前30位是哪些。所以这一个药物的话，脱离了剂量来谈它的功效也是耍流氓。所以一定要知道在足够浓度下它有怎么样的一个作用。这样子的话呢，结合君臣佐使的情况，结合那个主要的入血或入脏器的这个成分的重要性的情况，还有呢就是前期我们也要做很多的文献调研，就是可能我们也会做，比如说会建一个小分子库。我们其实很多时候你看到我们对于某些化合物它的个怎么样的作用，但前期我们往往是经过很大量的筛选的，就是筛选如果结合上一些就是有指导性的，比如说网络药理学，或者是刚才我说的一个量的和君臣佐使的问题的话呢，结合上这些我们就是优先等级排序先进行筛的话呢，可能会有重要性的这个化合物可能会这个比例相对比较高一点筛出来，但是绝大部分还是并不是那么的有效果的，所以筛是有一定比例的。那么只有你真正的这个筛之前，我们同时会结合文献调研，就是以往的研究当中是不是发现这一些化合物具有怎么样的一些作用，然后它的已有的这个研究 研究的结果有没有相关的提示，或者可不可能就是提示这个化合物在我们这样的一个体系当中有什么样的作用？当然不是说没有以往的研究基础就一定不是一个好药，不是这样子。但是有类似的研究基础的话，可能会更好的提示这个化合物的这个作用的效果。那么就是提示的这种预测到这种有可能有生物学作用的这样的一个是化合物的可能性会更大一点。所以综合的这些来研究，那其实也有很多的文献研究说，可能20个化合物它都很有抗炎作用，但实际上我们在我们的研究体系里面做了研究的话，可能会有非常明确的，比如说5、6个是非常明确的抗炎作用的，其他的有抗炎作用，但是区别并没有那么明显，效果并没有那么明显。如果是这样的话，我们也会后续在深入研究的过程中，因为时间、精力和金钱的各方面的原因，可能会侧重于研究那个生物学作用更明确的这些化合物的成分所以大概是这样子。
好，感谢您的回答。然后就是基于您刚刚提到的这一点，尤其您在讲的过程当中有提到，在基于传统的药方当中，我们要根据它的剂量，它用药的剂量来推断它在最后的汤剂当中的浓度，并且根据这个去判断它在整体的比如说抗炎作用当中所起到的作用的大小。这个我就想基于这个，就是进一步跟您交流一个问题，就是您认为就是在这个现在的运用现代的科学研究方法去尝试检测检验这个传统的医疗汤剂的方剂的这个指标当中，我们要多大程度上的奉行经典，我们要多大程度上的遵照它的这个东西来预测，或者说来期待它能够提供optimum clinical effect， 它有多大程度上能够给我们带来最佳的临床效果。然后如果我们比较经常能够如果我们现在的研究结论比较常见，就是经常能够发现我们跟传统经典研究当中的它的指标是相近的或者类似的话，您认为为什么我们现在能够重新发现跟这个传统研究当中重新形成共识呢？还是或者说，如果我们跟经典当中的指标不一样的话，您认为古人可能在哪些方面可能有一些谬误，或者说我们现在具有哪些优越性，使得我们可以超越他们之前的一些标准的制定呢？
其实这个角度来讲，古人呢，因为古人他往往是尊重着这个中医基层理论的这个指导和中药的配伍使用，包括他们有18反各种的这个禁忌的配伍，他们是通过了这个数千年的使用以后积累的经验来指导这样的临床的。那么的确有很多。我们不是说中药是万能的，但是的确有很多类的疾病当中，中药的使用是它的优势领域。它作用的，特别是像这个胃肠道等等的这个功能调控像这种代谢性的疾病包括我们现在的急性重症的这种感染性的疾病有些还的确是中医药的优势领域。那么古人的这个经验来说，主要是以传承了它临床效果比较良好的这样的一些方剂，比如非常有名的麻杏石甘汤。它是一个治疗流感的一个重要的方剂。那么现在应用的治疗新冠的这些方剂里面，很多比如说清肺排毒汤坏湿败毒方这些，它的底方都是麻杏石甘汤。所以这样子的话，也是印证了古人的一个提出的这样的一个治疗流感的经方是非常非常有用的，经得起考验的。我们其实很多，我刚才也一开始在那个 overview 介绍的时候有说到。我们的现代研究主要是2个作用。第一个作用呢，可能就是明确了解这个中药为什么有用，汤剂也好，化合物也好，它为什么有用，什么物质在发挥作用，通过哪些机制发挥作用，这是我们能够用现代科学手段参与进来的，这个叫还原论。还原论就是说你复杂的这个中药的研究体系，加上复杂的这个机体的反应，最终是有临床效果。那么 OK， 你告诉我到底谁发挥了什么作用，这个是还原论。但是还原论呢，不完全是中医药发 展的，当然它是重要的一个发展的一种手段，但不完全，因为你很难用还原论的这个方法来穷尽中药的这样的作用的机制。那这个过程中，我们就是要谨慎的探索，就我们要层层剥开它的作用的机制，并且慢慢的还要恢复到它的一个就是古人的智慧。我们说古人的智慧有时候还在于这个配伍啊，包括有效成分的这个，包括数量的比例啊。你刚才也提到了《君臣佐使》里面有些用药它的剂量也是有不同的呀，对吧？这个不传之秘嘛，中药的剂量。那么这些怎么来体现？不是简单的说 A 有什么作用 ，B 有什么作用就结束了的，是不是 A 加 B 加 C 可能作用效果更好？那这样子的话呢，就是我刚才也提到是一个小辅方的概念，你的有效成分以什么样的比例配合在一起，作用效果会更好。但这也只是就是现代中药发展的一个小分支啊，叫我们说小组方的问题。小组方的这个推进啊，这个当然是一个好的探索，但它还是要谨慎的对待，因为我们老祖宗留下来的很多值得继承的经验或者临床的有效的这个根据，还不完全能够保证被我们现有的科学手段能够很好的理解的，所以这个地方还是要就是在还原论在小辅方这样的一个理念下，并不是完全要摒弃或者取代我们原来的这个中药辅方的这个概念。
我理解，所以就是说，不仅我们一方面我们要通过还原，就是基于还原论的这样的怎么讲这样的站位吧，我们尝试去解释现有的经典当中的方剂当中的所发生的一些分子上正在发生的机制，这是一方面我们现在可以确认去做的东西，但另一方面我们要实现对于这种固有经典的超越。这个超越我们必须要谨慎的去做，像您说的，但是可能这个超越并不限于说，他说是什么就是什么，我们可以重新再一次 rediscover， 或者说重新，
因为其实我们还有很多，
您说，
对，还有很多未知的领域，比如说刚才也有老师提到了经络的问题，那么这个经络的具体的物质是什么？它是怎么发挥作用？这些我们其实还是有很多的空白的地方。因为中医的这个整个理论体系和它的这个发挥作用的这样的体系，跟西医是完全不同的。那么这个中西医结合也是在不断的探索中，用你来解释我，用我来解释你，可能有些地方也不能互相解释。但是这样子的话，我觉得是求同存异吧，能够互相利用一些好的手段，然后结合在一起。
明白。换言之，中医可能可以向西医提供一些新的认识人体的维度上面的
对整体论的一个概念
扩展，反而可能可以反哺给我们现在的草药学研究，因为我们对于人体有更多维度的掌握的情况下，就更有可能能够明确其中的机制。OK， 我明白了。那我最后再问您一个问题，就是也是帮助在场的各位同学，还在听的各位同学，谢谢大家待到这么晚。就是想跟您沟通一个，就是想跟您了解一个情况，就是如果在场的同学都是对于中草药研究有热情，大家都想要进入这个领域，而且大家都是未来有可能可以进入，比如说分子生物学、细胞生物学等等一系列这样的这个研究的这个途径上去的，大家都基本上都是本科或者研究生刚开始读，所以说大家都处于一个还在选择自己未来研究方向的阶段。那如果现在的大家想要进入到比如说中西医结合的草药研究这个领域当中，您觉得现在这个领域的研究机会是否很丰富？这个领域的科研的环境是什么样子的？然后包括如果大家作为现在的学生想要为这个领域做些什么，我们作为我们对大学生、研究生，我们有什么是我们可以做的吗？
我觉得很好啊，因为如果年轻人对这个领域有兴趣的话呢，我觉得是个非常非常值得鼓励的一件事情。因为其实中药的研究或者中西结合的研究，我个人认为它也是一种科学。就这种科学呢，就是可能会有一些就刚才提到的是整体论和还原论的问题，它可能是双方都要考虑的这样一个，就是还是具有很多的未知的一个新的科学领域。那这一个您刚才问我的是机会多不多，合不合适的机，那我觉得 大家也都知道习大大的这个执政这么多年啊，就是对我们国家的这个稳定和经济的发展是起到了很重要的作用。但是对中医药的研究领域的发展，我可以说它是一个这个决定性的 fundamental 的 substantial 的一个促进和一个推动的作用。所以在这样的一个政策的支持下，当然也在这个中医药的这么几千年的历史临床这个效果的见证下，中医是不是科学，这个已经不再是一个很值得讨论的一个问题了。那么大家因为也是在国外各个非常好的学校里面有这样的很好的一个学习的平台和机会，我希望大家能够就是师夷长之以制夷吧，也不光是这样子，就是说需要拥有一个开放的心态去学习更多的一些手段啊、方法呀、这个体系啊，包括一些理念啊，然后能够做一个能够把自己期待培养自己成为一个大科学家。什么叫大科学家的概念呢？就是说我学了比较多的知识，我的眼界比较宽，我感兴趣的内容比较多，但是我并不局限于我的已知的这些知识。我拥有一定的开放的心态，我愿意去拥抱一些未知，我愿意去积极的探索一些这个借助一些我已知的，或者说借助一些现在流行的或者未来有发展趋势的这样的一些技术手段，来解决一些真正有临床疗效或者真正值得研究的一些科学问题。那我觉得这个就会是一个非常好的一个探索和结合。那么中医药这个研究领域的话，周宗岩老师也在啊，也是我的非常优秀的同事。那个我是觉得，目前整体的这个研究人员的水平啊还不太够，就是大家还是处于自由探索或者自由发展阶段。当然这几年会很好，全国各个综合性大学的很多，比如说南京大学药学院什么像这种药科大学，他们这些人都开始研究中西医结合的问题。那真正能够贯彻中西医结合理论或者中医基础理论的这样的一个能够尊重中医的一个发展的规律的前提下面做有水平的基础研究，这一块我觉得这一些人群还是很少的，所以很需要这个高水平的人员的加入，并且我相信高水平人员的加入的话，它会比在综合性大学或者做纯生命科学研究领域更容易出彩。我们现在的话，国家的这个各个级别的人才计划，包括优青、节青，甚至全国的院士的评选，其实对中医药领域都是有非常好的倾斜的。那我想，对于年轻人来说，或许这是一个价格的洼地，很好的一个发展的一个机会。但是呢，最后我也想提醒大家，就是不要太功利。就是要做一个研究，一定是要有自己内在的驱动力和内在的兴趣和爱好的。就我不是为了晋升职称那么简单，我也不是为了这个拿几块钱的奖金那么简单，而是我想真正的搞清楚它为什么有作用，怎么发挥作用了，它这个中医理论到底好在什么地方，体现在哪里，哪些地方值得发展。然后我觉得这些是一个你的好奇心和你的热爱真正能够驱动你长久发展，在这个领域深耕，并且取得令人瞩目的成就的一个最核心的一个力量。
好的，好的，感谢您，感谢您。今天也耽误您的时间了。所有还在这个地方的同学们，我刚刚在咱们的这个 chat 里面再次发了我们这次峰会的这个相当于调查问卷，希望大家参与了今天峰会的全程的同学们能够帮助我们填写一下这个意见，然后让我们了解一下我们做的怎么样，包括未来的我们可以在哪些方面上面进行提升。也感谢郑老师，感谢刚刚参与的骆老师，以及现在已经离开的周老师感谢三位老师今天这个非常全面的分享，然后每个人也在自己的专业领域向我们展现了 中草药现阶段的研究是怎么样最大限度的与现代化的科研手段进行结合，并且尝试去用一些用一些现在学术界可以接受的语言，一些研究成果进行对于我们传统的经典方剂的疗效进行再解释的。我觉得这个非常感谢各位老师的分享。那我们今天的这个 sub panel， 我们herbal medicine 的 sub panel 就到这里。之后我们会将这个允许我们录像的2位老师的 presentation 的部分放到网上。这个我们到时候也会就是等我们完成了我们整个的这个峰会的上传之后，我们也会通知大家，因为我们有大家的邮箱，我们会到时候通知大家去哪里能够找到本次峰会的这个录像，包括剩下的2个 panel， 如果各位感兴趣，也可以到时候去那个地方，就是2个分会场，如果大家感兴趣，也可以去那个地方找到他们相应的 presentation 的录像。谢谢大家今天的参与。我们今天的这个峰会就到这里，感谢大家。
好，谢谢。
谢谢。
感谢大家的参与。